Hello everyone. Today I'm going to give a presentation for illustrating my research proposal. My name is Yichen, and my supervisor is Professor Vicky Dijkstra. The subject of my research is liming effects on soil microbial biomass and enzyme activity, a meta-analysis. In the next 10 minutes, I shall introduce my research proposal from 5 sections: the introduction, aims, research plan, expected outcome, and my research timeline. The first section is an introduction of the research topic. As we all know, healthy soil is important for agriculture system, even for the whole land ecosystems. Among different biological soil quality indicators, the microbial biomass and enzyme activity are two important evaluation indicators for assessing the soil quality and structure. They represent a part of soil function related to residue decomposition, nutrient transformation, and recycling, and the sequestration of soil organic carbon. Therefore, a favorable soil microclimate condition for soil microbial communities is necessary for the land ecosystem. However, acid soil will affect this function. Soil acidification is one of the most important problems in the agriculture system. It can be caused by both the nature and anthropogenic processes, acid rain, legume growth effects, and a large amount of fertilizer application. What's more, virus acid-causing substances like sulfuric acid, nitric acid, and ammonium salts, they all lead to decreases in soil pH. More importantly, this condition will not only affect the plant growth but also affect the microbial biomass and enzyme activities. Therefore, in order to solve this problem, liming is necessary. Since ancient Rome, lime has been applied to solve the problem of soil acidification. Even now, the application of lime is one of the most common and effective methods in dealing with soil acidity. In agriculture system, lime mainly comes from coastal dunes and crushed limestone. According to the report from British Survey of Fertilizer Practice, the most common lining material for arable crops and grassland is ground limestone. The second most applied material is the dolomite limestone. Through the releasing of calcium ion and magnesium ion, the hydrogen in soil can be neutralized. In this way, soil microbial community will be improved with more microbial biomass and a greater enzyme activity, and finally ameliorating the soil condition. In this project, I will use a meta-analysis to assess the effect of liming on microbial biomass and enzyme activity, and I will offer a quantitative analysis of overall response of microbial biomass and enzyme activity to effectiveness of liming across a wide variety of soil types and environmental conditions. After the introduction, the second section is my project aims and the hypothesis of the outcome. Firstly, through the meta-analysis research, I'm going to generate a quantitative estimate of liming effects on microbial biomass and enzyme activity. Secondly, I hope to figure out how the soil properties like soil texture, soil organic carbon content, and any other factors lead the result of liming. In the meantime, I hypothesize that lime treatment will increase both microbial biomass as well as the enzyme activity. But liming effects will also depend on rate of lime application, soil type, and environment conditions. After setting the research targets, a corresponding research plan is necessary, and the third section is my research plan. In the first step, I need to collect the data for coming meta-analysis. I'm going to use the Web of Science database to find out some peer-reviewed scientific articles. Here is the homepage of the database, and the initial search queries are here. After initial search, Papers published before January 2022 will be screened, and they will be selected based on the following criteria. At first, the experiment in the paper should have at least 2 replications, and next, the paper should involve the information of microbial biomass or enzyme activity, and then the experiment duration must clear and with the specific liming instructions, such as the amount of lime used and the type of lime. Finally, in order to make good use of these articles, the paper should contain some necessary experimental data, like the mean value, the standard deviation, or the standard error. What's more, I will further collect other auxiliary data on soil parameters, including soil pH, soil texture, soil organic carbon and nitrogen content, crop type, and some information about whether the study was done in the field or in glasshouse or in laboratory incubations, when it will be conducted in the field, as well as the location and environment conditions. After the first step of Section 3, the second part is to analyze the data collected before. As for statistical analysis, I will use meta-analysis method. It has been widely used in agriculture and soil science and has contributed significantly to quantitative analysis about biological issues. In this meta-analysis, the effect of liming on microbial biomass and enzyme activity will be quantified as a response ratio between these two microbial parameters in liming and control treatment, with the following equation. In the first equation, XL and XC are the mean value of microbial biomass and enzyme activity in 2 different treatment groups, the light treatment and the no-light treatment. Apart from that, the variance of each study response ratio is calculated through this equation. In this equation, NL and NC are the number of replicates for liming and control treatment. The SDL and SDC are the standard deviation for liming and control treatments. Moreover, to analyze the final effect of liming and microbial biomass and enzyme activity, I will make use of our software package to calculate the mean effect size and 95% confidence intervals. After these calculations, if the result of confidence intervals are not overlap 0, the response of liming to microbial biomass and enzyme activity is significant. And finally, I will further assess to that degree the liming effect depending on liming amount and type, crop type, soil parameters, and environmental conditions. In the next section, I'd like to talk about some expected outcomes of my research. Firstly, I'm looking forward to finding out at least 15 useful experimental papers. This will determine the quality of my research. The more suitable articles I can find, the more reliable research result will be. Secondly, I hope I can find that liming effect is indeed positive for soil microclimate organisms, even if there will be some differences because of various experimental conditions. The total effect of liming on microbial biomass and enzyme activity should be significant. At last, the final section of my presentation is my research timeline table. In terms of my timeline, I will finish my research at the end of April 2022. During the whole time, I have set up some phased tasks to ensure the smooth progress of my research, and I think I can also complete my task in time according to my plan. Maybe there will be some changes happen, but I think they could not affect my processes. This is the end of my presentation. Thank you for your listening. and there is some relevant bibliography. Yeah.